Okay, hello to everybody. Now we are going to discuss chapter number 15. This chapter is from infection chapter. So every week we will be also discussing one infection chapter. So let me go into slideshow mode. So in this chapter, 15 basically talks about how to diagnose the infections. So basically, so if we have some clinical sample, then how to identify from the clinical sample which kind of microorganism is there in the clinical sample. So basically that's what we're going to study in this chapter. So there are three ways by which we can identify the bacteria in the patient samples, or if we have some sample from the nature. So for that also we follow the same techniques. So first of all is phenotypic, then is immunologic, and then is genotypic. Okay, in the phenotypic part, what we do, we just consider the macroscopic and the microscopic morphology of the bacteria, and we also go for physiology and the biochemistry. So in the phenotypic, we go and look for the morphology of the colonies, which we can see with our eyes, that's macroscopic, or we can take the bacteria from the colonies, we can take some part of the little speck from the colony and make a smear out of that, put it on the slide. We can stain the slide, we can stain the smear and just put the cover slip, and we can look under the microscope. And we can also perform various biochemical tests which can show us what kind of enzymes the specific microorganism or the bacteria has, and on the basis of that we can identify the bacteria, what kind of bacteria is there in the clinical sample. Now the immunologic way is by using the detection of antibodies in the patient sample. If the patient is exposed to or is infected with some specific kind of the bacteria, then that patient should have the antibodies in their serum, because the antibodies, the formation of antibodies is a part of our immune system. So when we are exposed to some kind of infection, our body produces the antibodies, and these antibodies are very specific to the antigen because they are produced in response to the antigen, and the antigens, they are present on the bacterial and these antigens are part of the bacteria. So these antibodies are very specific. So we can also identify um, the, the the bacteria which is causing the infection by the presence of the antibodies. And the last one, and which is getting very good way of detecting or identifying the microorganism, is genotypic way. So this is really becoming very popular these days. Genetic techniques are becoming very popular these days because they're very reliable, plus they can be done in really shorter time because they do not require the culturing of any bacteria. Usually the culturing of the bacteria requires something like 18 to 24 hours, or sometimes some bacteria even require longer time period, some 48 hours or more than 48 hours. So by this technique, by this way, we can do the detection really fast. And also there are some microorganisms, some bacteria, which we cannot culture them in the lab, or they're very difficult to culture, and some of them we cannot culture. So in that scenario also, genotypic techniques or the genetic techniques are very useful. So these are the different ways how we can identify the bacteria, and we can come to know the infection is caused by which specific bacteria. So this table, this chart basically shows the same thing. So methods of microbial identification. First is phenotypic, which is on the basis of microbes, microscopic and macroscopic morphology. We can look the morphology of the colonies, which is macroscopic, or we can take the bacterial cells, make a smear, stain them, and look under the microscope. That's microscopic morphology. their physiology or their biochemical properties. There are various tests which can be performed in the lab by which we can identify the microorganisms. Immunological, as I said, they are on the basis of the antibodies which are formed against the antigens on the microbes. So if we can detect these antibodies, we can identify the bacteria. And the last one is genotypic, and that is on the basis of the analysis of microbes DNA or RNA. So on the basis of the analysis of the DNA or RNA of the microorganism, we can identify the microorganisms. Okay, so basically we have already talked about just a repetition. So phenotypic ways is on the basis of the it is on the basis of the observation of the traits that an organism is expressing. So that involves basically, as I said, we can examine microbes' appearance and its behavior also, and it's on the basis of what kind of enzymes they have, what kind of physical conditions they live in, or the antibiotics they're susceptible to, or we can also look for the chemical composition of the walls. Basically, we can do gram staining, and we have studied in chapter number 2 about the gram staining. It's a differential stain. It can differentiate between gram positive and the gram negative bacteria. Gram positive bacteria, they have a thick cell wall, and gram negative bacteria, they have the thin cell wall. Immunological, it's also a very good way of identification, because this is on the basis of the presence of the antibodies in the serum of the patients. And these antibodies, as I said, they are formed in response to the antigens which are present in the microbes. So they are very specific to the antigen which is present on the which is a part of the microbes. And genotypic or the genetic techniques, as I said, they are getting very popular these days, because for these methods, culturing of the microorganisms is not required. They're quite fast, and they can give us they're very genuine way and very authentic way of identification of the bacteria. And they are also useful for those bacteria or those microorganisms which we cannot culture in the lab. Okay, so in this whole step of identification of microorganisms, the first most important step is the specimen collection. The specimen has to be collected in a very, very good way, because if the specimen is not collected in a good way and it is contaminated, then it is very difficult to identify the bacteria. So success of identification and treatment, because first of all we are going to identify the bacteria which is present in the clinical sample, and only on the basis of that treatment of the patient will start. So the success of identification and treatment depends on how specimens are collected, handled, stored, and cultured. So aseptic technique is very, very important, because if you are not collecting the specimen in a proper way, and your specimen is getting contaminated either from the environment or from the patient, then you do not have a good specimen. Okay, so care should be taken with samples that contain resident microbiota, because when you collect the sample, there will be some resident microbiota also, so we have to be careful with that also. So care should be taken with samples that contain resident microbiota. So only the infected site should be sampled and not the surrounding area. So we have to be very careful with that we are taking the sample from the infected site and not from the surrounding areas, because as we know that microorganisms, they are ubiquitous, they're present everywhere. They're on our skin, they're part of our body, they're doing good things for us. But if we have to take a sample, we want to take the sample from the infected site. So this slide shows you the different samples we take. Like, it can be blood sample, urine sample, it can be skin swab, it can be fecal sample, it can be vaginal sample, sputum, saliva, various samples can be taken. It all depends on the kind of disease somebody is having, or or it depends on the kind of infection the person is having. And when we take the sample, we can start with the phenotypic methods. We can take the sample and we can take the sample on the slide, and we can stain. We can make a smear, and we can stain it. We can do gram staining. We can do acid fast staining. So I'm not going to repeat these because we have already talked about what is gram staining, what is acid fast stain in chapter number 2. So basically we can have the sample, and we can stain the sample, and we can look under the microscope. Or we can also take the sample and use it as an inoculum, and we can also inoculate those specimens on the different kinds of medium, which means we can culture the bacteria which is there in the specimen on these mediums. And we have already talked about, we can, for this purpose, we can use selective medium. So as you already know that the selective medium, this also part is done in chapter number 2. Selective medium is that medium which is going to let only a selected microorganisms to grow. Like some mediums will not let gram positive bacteria to grow, or some mediums will not get gram negative bacteria to grow on them, because they have some agents in them which inhibit the growth of some specific kind of the microorganisms. And we can also use differential medium. Differential medium can let multiple type of microorganisms to grow on them, but it will let the multiple kinds of microorganisms to grow on them, but those different kinds of microorganisms, they will show different characteristics, and on the basis of the different characteristics, which can be in the form of different morphology of the colony or different colors of the colonies, we can actually distinguish between different kinds of the microorganisms. So we can this is all coming under the phenotypic techniques. So we can also grow these, we can culture the bacteria which is there in the clinical sample on these kind of medium, and it can help us in the identification of the bacteria. So this is the flow chart. Basically it shows you how we can proceed with the microscopic analysis, or microscopic analysis, and with the biochemical analysis. So this is called dichotomous key, because here you can see like you can do the gram staining, and on the basis of the gram staining you can see here that if like in the this will be more explained in the lab. Basically, like the bacteria, they can be rod shaped. There are different shapes of the bacteria. They can be like spherical, like the round, they're called cocci. And then there can be some bacteria which can be in the form of rods, or some bacteria, they are like spirals. So the cocci, suppose you did the gram staining, and under the microscope on the slide you found that all the bacteria, they are like round spheres, so they are cocci. So suppose you got gram positive cocci. So then after that, what you're going to do, you can do various biochemical tests. One of the biochemical tests is called catalase test. It is basic catalase is kind of an enzyme. So if that comes positive, okay, and then so if it under the cocci, if they are gram positive, so they're called gram positive cocci. Then you do the various biochemical tests, like if it is catalase positive, and also if these cocci they are present in irregular clusters like tetrads. So if these cocci are present in clusters, then you can check if they are aerobic or anaerobic. If they are aerobic, so you can see how the scheme is going. So first of all, they are cocci, then they are gram positive, then they are catalase positive, and if then they are clustered in these clusters. And if they are aerobic, then we can identify them as micrococcus. But if we have cocci, they are gram positive and they are in the form of these clusters, but they are facultative anaerobes. Facultative anaerobes means they can live in the presence of oxygen. They are facultative anaerobic. So then we can say that they may be then they are staphylococcus or planococcus. So this is the way we perform the test. So this is microscopical test, and then we do some biochemical test, and on the basis of that we can identify the bacteria. Suppose here, as we have done here, like if suppose a cocci, but in the gram staining they are gram negative, which means they are showing pink color. Okay, so they are gram negative cocci. So how to proceed? Then you can see the different biochemical characteristics. Suppose they are aerobic, they are oxidase. This is oxidase is another kind of a test. They are oxidase positive. They are also catalase positive. then we can call them, we can say that, okay, they can be Neisseria, it's called Brenhemella, or Morazella. So they can be these microorganisms. Then there are further tests to identify which out of these three is there. Okay, and if again on this side, cocci, gram negative, suppose they're anaerobic, oxidase negative, catalase also negative, oxidase, catalase, these are both enzymes. If they are all they, so if they are anaerobic and all these are negative, then we can say that this microorganism may be Veillonella. Okay, so this is the way how we do the identifications. So as we have just discussed, biochemical testing is basically on the basis of the presence of some of the enzymes in the bacteria. So we have already seen in that scheme there are various enzymes like oxidase, catalase. So on the basis of that we can identify. Basically, for the biochemical test, the bacteria is cultured in the medium with a specific substrate. Then on the basis of the enzyme which is present in the bacteria, they're going to break this substrate and the product will be formed. And because we have added some color indications in the medium, so when the product is formed, it is going to give some specific color to the medium because of the presence of these color indicators. They are basically different kinds of color indicators. On the basis of that we can tell if this enzyme is present or not. Okay, so microbial expression of the enzyme is made visible by a colored dye. So if there is no coloration means it lacks the enzyme for utilizing the substrate. So this is a scheme. So if you have an unknown microbe, so you are going to grow this unknown microbe in some medium which has a substrate. Okay, if the enzyme is present in the microbe, it's going to break this substrate and the product will be formed and the color will change. So it means that, so the color of the medium will change. It means the microbe is positive for the presence of the enzyme. Suppose the enzyme is absent, so no product will be formed, no color change. So it means that the microbe is absent, the microbe doesn't have that enzyme. Okay. Now, what are the drawbacks of phenotypic methods? Yeah, they take very long time because for these tests, the microbe has to be cultured, and it usually takes a minimum of 18 to 24 hours. Okay, and maybe there can be some infectious diseases which are caused by non-culturable organisms. You cannot maybe you cannot culture some of the organisms. So in that case, if you cannot culture the organism, you cannot use these kind of methods. So what are the other ways by which we can identify the microorganisms? Another ways are immunological methods. As I told you already, immunological methods basically work on the they depend on the presence of the antibodies in the clinical sample. So if a patient is exposed or is infected with some bacteria or a microbe, then these microbes, they have antigens in them or on their surface, and our body reacts to these. Our body's immune system actually defends us from all the infections. And how does it defend? It defends us by producing the antibodies. So these antibodies, which are produced by our own immune system in response to the infection, they are very specific to that specific agent which is causing that infection, or the microbe which is causing, or the bacteria which is causing the infection. Okay, so it all depends on the presence of the antibodies, and those antibodies are specific to that microbe. So this is based on the principle that antibodies have extreme specificity for the antigen. Okay, this is very, very important point. And how can we detect the presence of antibodies? That is done by the visualization of the interaction of antigens and antibodies, macroscopically or microscopically. So we can basically look for the interaction of antigens and antibodies, and we can uh, detect if the antigens and antibodies are interacting. So the antigen and antibody will interact only if the antibody is specific to that antigen. Okay, so when they interact, they form a kind of a clump, and which can be seen with the eyes. You can just see them either under the microscope, or you can see them with your naked eyes. So the interaction of antigens and antibodies is when they interact, they can form a kind of a clump, and we can see them. So you can see here, so here if you see in this slide, there are two questions. Does the patient make antibodies towards the microbe? So this is patient serum. Okay, so serum can be prepared from the blood of the patient. Now we want to see if this patient is infected with some specific kind of a microbe. So somebody is sick, suppose somebody is infected. Now we want to check if that patient is suffering from which bacteria. So you can take the serum from the patient, okay, and you can because in the serum there are antibodies. So you can see like the antibodies are like this, okay, and you can have a prepared known microbial antigen. Suppose the suspicion is maybe this person is suffering from maybe staphylococcus, or I'm just making up streptococcus or something. So you can take the prepared antigens from a specific infectious agent, and you can mix that with the serum of the patient. Okay, so if the antigen and antibody interact, okay, if the antigen and antibody interact, because on the antibody there is a specific site which is called antigen binding site. Okay, so you can see here the antibodies are usually Y shaped. So you can see this is the structure of the antibody. So you can see how the structure is. So if the antibody is specific for the antigen, then the antigen and antibody, they're going to interact with each other. If they're not specific, they're not going to interact. So here if you can see, this is the serum from the patient, and these are the antigens from the microbe. So you can see here they are not interacting. So this is the antigen, this is the antibody. They are not interacting. So which means that this patient is not suffering, or is this patient is not infected with this specific microbe. Okay, but in this case here, if you see, this is the antibody and this is the antigen, they are interacting with each other. So if they interact and they form this kind of a complex, then it is sure that this patient is suffering from this kind of an infection. Now if you have to identify suppose this is a second question. So if you want to identify the microbe, okay, so what you can do in this is, suppose you have this was taking the serum from the patient. Now suppose you have cultured the you have taken the specimen and you have cultured it on the plate. Okay, so these are the colonies growing. Now you want to know what kind of microorganism it is, right? So it's just a colony growing. So by looking at the colony, we can't say. We can have some ideas. Maybe this is this, maybe this is E. coli, maybe this is staph, or it's Chpt, but we don't know for sure. So how we can take the help of immunological methods and identify the bacteria or the microorganism. Again, we can make use of the same principle of antigen antibody reaction. So you can have antibodies of known identity. Suppose these are the antibodies against S. aureus. S. aureus is Staphylococcus aureus, and this is the bacteria. This is the colony of the bacteria. We don't know which microbe it is. So you can take these cells, okay, these bacterial cells, they will have those antigens on them. Okay, so this is the bacterial cell and these are the antigens on them, on the microbe, and you can mix them with the serum. Okay, so these are the antibodies. Okay, these are the antibodies against Staphylococcus aureus. Now suppose these antibodies bind to this microbe. Okay, if these antibodies bind to this microbe, then we know that this microbe is Staphylococcus aureus. But if they do not bind, then it is not Staphylococcus aureus. So you can see here what is shown here. This is the microbe and these are the antigens on the surface of the microbe, and these are antibodies. So they are not interacting with each other, which means that this colony is not from Staphylococcus aureus. This bacteria is Staphylococcus aureus. But on this side you can see this is the bacterium, okay, and these are the antigens on them, and you can see all these blue antibodies which they're shown with blue color. They're all interacting with these antigens, okay, you can see here. So which means that when they're interacting, it means this bacteria is Staphylococcus aureus. So this is the way we can make use of immunological for identifying the microbes or the bacteria. Okay, antibody to a specific of an antigen. So there can be various kinds of antigens on the bacteria, and on each specific antigen there is a specific site on the antigen which we call it as an epitope, and the antibody binds on a specific epitopes. So basically as I said, antibodies are very specific to the antigens. They bind on a specific site or a epitope on the antigen. So when they interact, because every antigen is actually bound by different so you can see here, like this is one microbial cell, right, and it has these antigens on its surface, and you can see how many antibodies are binding to one cell. So when the antibodies bind to the antigen, they can form a kind of a clump, and we can see those clump. On the basis of the clumping, we can say if the antibodies are specific for the antigen or not. So if the antigens and antibodies are suspended in a solution, under the right circumstances, binding can be seen as clumps in the solution. So there are two kinds of reactions by which we can actually see the clumping, or we can actually say that there are two kinds of reactions by which we can see the interactions of antigens and antibodies, and those two kinds of reactions are called agglutination and precipitation reactions. Okay, in both reactions, one antigen is interlinked by several antibodies, as I showed you earlier also. So I just showed you one antigen bound by different antibodies. Even one antigen can be interlinked by several antibodies, and that form a insoluble aggregate that settles out in solution. Okay, so that is the way how antigens and antibodies, they interact with each other, and they form these insoluble aggregates. So this is the way how antigen and antibody, they interact with each other. So there are two types. One is called agglutination and precipitation. So what's the difference between them? So the difference between agglutination and precipitation is the antigen which is examined in precipitation is a solid molecule, okay? And it precipitates in separate medium because it is a solid molecule. And the agglutination is a whole set of organisms such as red blood cell bacteria displaying the surface antigens. So in this case, in the case of agglutination, it's very easy to see the interaction of antigens and antibodies in the form of visible clumps. Okay, because antigens are whole cells or organisms. There's a one kind of a agglutination reaction which we call it as a Biol reaction. It's a kind of a agglutination reaction, and it is used for the diagnosis of rickettsial infections. And also agglutination is used for not for diagnosing, but for the testing of the blood types. What your blood type is? Are you A plus, B plus, or your O negative, O positive, AB negative, AB plus positive? So that can also be the blood type. What kind of blood type you have, that can also be tested with the help of agglutination reactions. So you can see here, this picture is showing you agglutination reaction. So you can see this is the control. So and you can see here that agglutination is taking place, and you can see these clumps forming. So here such a big clump is there. So this is showing you the interaction of antigens and antibodies. So the plus has been given to you because it's a very big clump, and then the clump is smaller here as compared. So 2 pluses, and there is some clump formation here, 1 plus, and here no clumping, so negative. So in this plate also, it's shown you how the antigen and antibody, they're interacting, and here you can see agglutinated mat. You can see these are the antigens, and these Y-shaped structures are the antibodies. You can see how this antibody is interacting with these cells. Okay, so you can see how this antibody is interacting with these cells, and how the in this case antibodies and the which are part of this, there is take place in this cell. Okay, the last method to discuss is algorithms. So as I said also, they are very authentic way of detecting or identifying which they are very authentic way of identifying the microbe or the bacteria which is causing infection. One of the technique which is very predominantly used these days in diagnostic labs is called PCR, polymerase chain reaction. So this is the technique by which DNA can be amplified within few hours. So if infected with some bacteria, making use of this technique can basically take your sample, and it can amplify the DNA or the RNA which is present, the DNA or RNA of the microbe. So PCR results in the, as I said, PCR is a way by which the amplification of DNA can take place. So PCR results in the production of numerous identical copies of DNA or RNA within hours. Okay, so on the basis of that, we can identify the causative agent of the infection. And then there is another kind of PCR called real-time PCR. So basically, this real-time PCR uses fluorescent labeling. DNA is getting amplified. There are some fluorescent markers also involved in this kind of amplification. So the level of fluorescence is measured in the real time as the reaction is running, and we can see the amplification of DNA taking place in the real time. This method involves the fluorescent labeling. So more and more fluorescence, the level of the fluorescence, which means more and more DNA is getting amplified. This is fully automated and faster than the traditional PCR. And the last technique is called hybridization. So basically, this technique identify the microorganism by analyzing the segments of its genetic material. So basically, if some microbe is infecting you, so by this technique, the analysis of the genetic material of the agent which is causing the infection is done, and this technique involves some probes. So first of all, what are probes? So they are small segments of DNA or RNA, and they are to be complementary to specific sequences of the nucleic acid which is isolated from the microbe. So basically what is done in this technique is the DNA, the genetic material from the infectious agent is analyzed, and it is basically hybridized with some probes. And those, what are probes? They're small segments of DNA and RNA, and if they are complementary to the genetic material of the microbe, then they are going to go and bind on them. If they are not complementary, they are not going to bind on the genetic material of the microbe. So so there are different probes, like if it's just like the way I showed you for the immunological methods also, like you have a specific kind of microbe, similarly you have the probes for different kind of probes. Material, then we can say this person is infected of microbe. And the binding of the probe on the genetic material of the microbe which is isolated from the clinical sample is on the basis of the hybridization. Okay, and hybridization is on the basis of the if the probe complementary sequences, it will go and bind only to the complementary sequences in the genetic material of the microbe which is present in the clinical sample. And these probes, they are fluorescently labeled to an enzyme, and that enzyme is going to trigger a color change when hybridization occur. Okay, so the probes, they are fluorescently labeled to an enzyme, and if the hybridization takes place, a color change will take place, and that will let us know if the hybridization is taking place. If the hybridization is taking place, it means there is a complementation of the probe with the genetic material which has been isolated from the clinical sample, and that indicates that the patient from whom the clinical sample is taken, that patient is suffering from this specific microbe. I hope it's clear. And again, if you guys have any question, please let me know.